Let's come to the immunology. As we learn now in the part 1 about MHC class 1 and class 2 molecules. MHC class 1 and class 2 molecules. Here I'll remember you once again, MHC class 1 molecules are present in all nucleated cells. When the cell is infected, the nucleated cells is called altered self-cell. They present the particular microbe to the T cytotoxic cells, which leads to cell-mediated immunity. So now we are talking about in the immunology part 2 about the possibilities of the cell-mediated immunity. One of the possibilities shown now, that MHC, major histocompatible complex molecules, which are explained in the part 1, how do they play a role in the cell-mediated immunity. We say MHC class 1 molecules containing altered self-cell present it to the special kind of cells called T cytotoxic cells, which leads to the cell-mediated immunity. Let us see that possibility in the cell-mediated immunity, how it actually happens. So in the diagram, let us focus only on that MHC class 1 molecules we said now. See the first one, altered self-cell, ASC is given here. Altered self-cell, a cell which contain nucleus, a normal body cell we are talking about. When this particular cell is infected with a microbe and this cell is having MHC class 1 type of molecules on the cell membrane, when this particular cell is infected, we consider the cell as altered self-cell. That in the diagram we see that endogenous antigen, that means microbe entered in the body, in the cell. Then with the help of MHC class 1 molecules, MHC class 1 molecules, there you can see the MHC class 1 molecules, the cell is infected and it was named as ASC, that means altered self-cell. And now this particular infected cell is presenting, you see here the TC cell. What is TC cell? T cytotoxic cell. When T cytotoxic cells interacted with the altered self-cell, the such an interaction you can see here. Interaction is possible by a special kind of communication, we say interleukins. We discuss about the interleukins in the further. And what this T cytotoxic cell is doing, we learned earlier, they are responsible for cell-mediated immunity that we are now observing here. The T cytotoxic cell immediately will differentiate into two types of cells, like the cytotoxic T lymphocytes that you can see here, and a memory T cytotoxic cell which will remember that particular microbe for future references when it comes back again to kill that particular microbe. So memory T cytotoxic cell and effector. Then what effector is going to do? The effector cytotoxic lymphocyte on infected cell, they release perforins. There you can see the perforin. Perforation means making hole. So the perforins makes the hole on the infected cell. Our said altered cell is the infected cell. So perforins are making the hole. So there you can see that perforins released by the T cytotoxic cell makes the hole on the infected cell, and through the hole granzymes are released. Through the hole the granzymes are released, and the granzymes are released into the infected cell. The granzymes which are entered in the infected cell promotes the release of caspases. It's a special kind of protein, caspases. The caspases is going to promote the particular infected cell to die themselves. That means cell suicide we are talking about. Apoptosis, we say that process as apoptosis, programmed cell death. Programmed cell death. So now the infected cell is dying. So this is such kind of immunity we say cell-mediated immunity. Let us focus here on the cell-mediated immunity which is possible by T cytotoxic cells given in the box. Let me once again, a cell with MHC class 1, when it is infected, it is called altered self cell. Here you can see ASC, endogenous antigens, that means the antigens which can microbe which can enter into the cell, we say endogenous antigens. So then next, what happened? With the help of MHC class 1 molecules, they are going to submit this to the T cytotoxic cell. That activated T cytotoxic cell is differentiated into effector, cytotoxic lymphocyte and memory T lymphocytes. Memory T cytotoxic lymphocytes for the further use. At present, the effector cytotoxic lymphocyte who is participating in the cell-mediated immunity now release perforins which make the holes in the cell, and through the perforins the granzymes are released, and the granzymes entered in the infected cell promote a special kind of protein, caspases. Caspases are going to promote as process we say apoptosis, programmed cell death. The apoptosis which makes the cell to die itself, we call such kind of immunity with the help of T cytotoxic cells as cell-mediated immunity. Cell-mediated immunity. Okay, let us see the another type of cell-mediated immunity. Now we are talking about MHC. I'll remember you once again that MHC class 2 molecules, the possibility of MHC class 2 molecules in the cell-mediated immunity. We said now earlier in the first part, MHC class 2 molecules are of two types that present in the APC. We say they are called antigen presenting cell. The antigen presenting cells like dendritic cells, macrophages and some B lymphocytes also play this act. We call them as antigen presenting cells. There you can see this particular antigen presenting cell. MHC class 2 molecules, antigen presenting cells are present in the MHC class 2 molecules will manage the exogenous antigens. That means that antigens which are moving in the body fluids. You see, we are talking about macrophages here. I said there are many antigen presenting cells. Now I am taking an example of macrophages. See in the diagram, first diagram, that macrophages, you see that exogenous antigens are engulfed by the macrophages. They are processed, and that means cutting into the pieces. And processed microbe, we consider as antigen, is presented here, you see, MHC class 2 molecules. MHC class 2 molecules are presenting these to the T helper cells. Earlier we discussed this particular APC is generally present the microbe or antigen to the T helper cells. What T helper cells can do? That you see here, the T helper cells are submitting this particular microbe containing to the T cytotoxic cells, and another possibility of cell-mediated immunity. The T cytotoxic cells then differentiate into T effector cells and T memory cells for future, and effector cells act on that particular exogenous antigens, and that leads to cell-mediated immunity. So, let us see the possibilities of cell-mediated immunity again. Cell-mediated immunity, the possibilities of cell-mediated immunity. The one possibility we said with altered self cell. The altered self cells managing endogenous antigens are presenting to the T cytotoxic cells, and they differentiated into T effector and T memory cells. Effector cells act on that infected cells by releasing the perforins, and followed by granzymes enter into the cell, promote the caspases for the process called apoptosis. The infected cell will be destructed themselves. We call them as programmed cell death. We call such kind of immunity as cell-mediated immunity. This is the first possibility. The another possibility. The second possibility is with antigen presenting cells. Antigen presenting cells. The second possibility is with antigen presenting cells. These particular antigen presenting cells are releasing or managing with exogenous antigens. They are having MHC class 1 molecules. They present the antigen to the T helper cell. The T helper cells might they are going to present these to the T cytotoxic cells, the cytotoxic cells. We again differentiated into T memory cells and T effector cells, and effector cells are going to destroying the microbe with the process called cell mediated immunity. So this video focused on the cell mediated immunity, which is possible by T cytotoxic cells and the collecting microbe from either APCs, antigen presenting cells, or antigen altered cells. Antigen presenting cells via T helper cells and altered cells are responsible to present the microbe to the T cytotoxic cells, and then now the T cytotoxic cells are making this cell mediated immunity. Thank you. In the further classes, we discuss about the humoral immunity and the other issues of immunology. Thank you. And the further videos and for assignments and question papers with respect to the biology, you can download from Neet Srikanth, neetsrikanth.blogspot.com. Thank you.